Next one up, Lungegårdsvann again, in a different version. Go ahead.
Yeah, so this is John Sai, and I'm Daniel Selak, and our group checked the water quality in Store Lungegårdsvann, which is a semi-enclosed bay connected to Puttefjord. Water quality is checked by measuring the concentration of TCB. TCB is a thermal-tolerant coliform bacteria, which lives in animal intestines, and this group includes some pathogens. The TCB dies within hours or days if they get released into the natural environment, but a high TCB concentration indicates that fecal matter has been recently been released into the water. These are the EU directive standards, the TCB concentration corresponding to qualitative rating. So kids might get sick depending on where the water comes from. Not all the places have the same concentration. Usually you will get diarrhea if you swallow the water, but there are also a huge list of many other diseases you might get, and in some most extreme cases you might die. This is about 100 milliliter of water, and at site 1 we found a pretty large amount of number. I'm not going to reveal it now, but just think about this amount of water for so we can ask, is SOV suitable for swimming? Are there any correlation between tide and TCB concentration, or precipitation and TCB concentration? What about physical parameters like temperature, pH, and oxygen? Do they have any effects on TCB concentration?
So how did we find the answers to those questions? Before the sampling day, we discussed with group 3 members, and we analyzed the data from previous report, and we visited the sampling location. Then we make a design. This map shows SLV, and we choose 7 sites for our sampling. Near site 1, this place is a fire station, and between site 1 and site 2 is LLV outlet. Site 4 is a freshwater outlet. Between site 5 and site 6, Bergen commuter want to make this place to be a beach in the future. Site 7 is the dock. Here is a narrow place. Water exchange here is different. And here is a kindergarten. You can imagine that children may be walking around this area, and maybe in the summer they want to swim in this area. And here is a feeding place filled with a lot of rocks, sand, and people want to build something on this place. And you can see the red lines here is all the sewage pipes under the water. And these 2 places is discharge water point for the private use. And almost the site is the same as last year. We want to compare the result with them.
And so this is water sampling carried out. This is at site 1. And there you can see Maya collecting TCB, which was done at a depth of 1 meter. Here is the physical parameters being measured, which is temperature, conductivity, pH, and oxygen. Phosphorus sampling at site 4. Phosphorus was also sampled at 1 meter, but here we sampled at the surface because the depth was just 0.5 meters. Here we can see Ryan and Sean measuring with the cohort sampling the biodiversity. This square is 40 by 40 centimeters, and the small squares are 10 by 10 centimeters. We measured at the intervals of 2 meters. You can see the red dot, and this line is 10 meters. Ryan writes down the corresponding percentage, which is being approximate percentage which is being counted in the squares, writing it on the paper.
This shows the TCB result for recent 2 years, and you can see that most of the result shows just above last year, just above 1,000 CFU. It's not a specific number, so this year we asked the lab to dilute the water, and we got the exact number. Apparently, we can find that at set 1, we got the highest TCB result. And you can imagine that in this glass of water, there contains 7,500 TCB CFU from animal intestines. And if children drink this kind of water, they will get sick. And all the results give us the information that the water quality in this area is very bad. And this is the biodiversity result. Channel index reflects the different species in one community and the evenness of the distribution. And you can see that all the results for these 2 years is lower than 2. That means the biodiversity in this area is bad. And for example, at set 4, we got the highest channel index for this year, but we only found 5 species. The most one is this one, polychaeter. We found 7 polychaeter at this set. And we found other abundant species around this area is here. In this one is maxillopoda. But we only found this species at set 5. So the channel index result is 0. When we compare the 4 years result, we don't find any improvement for this year. The other important result we found is the precipitation have the positive correlation with TCB concentration. The precipitation data we got from Bergen Communer, and we compared 3 hours average, 24 hours average, and 1 week average precipitation data with TCB concentration. And then the 24 hours average shows the most positive correlation with TCB concentration. The main reason is that when high precipitation will make the runoff bring TCB into the water, and the heavy rain will make the sewage pipes overflow.
So the tide data didn't show any significant correlation with the TCB. That was at second glance. There are also small sample sizes with high variation. And as you can see here, we only sampled at high tide in a bunch of samples at that top. And we should actually sample at from low tide to high tide, even as you can see the red lines. So this is garbage and contamination examples from the three sites. Site 1, we had a lot of adult magazines. And also a lot of shoes and just garbage. This is our park area, so a lot of people is spending their time right there. And between site 2 and 3, there is a building site. So this it's also standing close to the water, so it is a contamination source. At site 4, we saw a street sweeping car being washed behind that wall. There was a hole in the wall where the wastewater was pouring out into the river. So the conclusion is that the TCB concentration are of course too high, especially on site 1. There are a positive correlation between TCB and precipitation. Channel index show low biodiversity. All the numbers were below 2. There is no correlation with physical parameters, that is temperature, pH, and oxygen. The number of samples at each site is too small. The maximum number of replicates of TCB were 3. And all the others was about 1, and phosphorus maximum was 2. So maybe we should do measurements at more sites around the SOE to get a clearer and more broader picture of the status of the TCB concentration. So the private pipes between site 7 and 5 are going to be removed soon. We should also check for more leaky pipes because they are pretty old. The Bergen community should also do a regular cleanup of garbage, especially at site 1, and also syringes at the rain hut between site 3 and 4. There are also a lot of feces in that rain hut. What about creating a database for better data access, like so that we can discern between useful data and faulty data? Maybe a internet forum for news and updates about how it goes with that checking for leaky pipes and regular cleanups and private pipe removal, and also a general information about the progress of fixing the water quality and the surrounding areas of SLB. Tack.